Hey everyone, in this video I am going to describe how the virus HIV-1 is reverse transcribed so it can be integrated into a genome. So HIV-1 is a single-stranded RNA virus, but it integrates into the genome. In order for it to do that, it needs to turn its single-stranded RNA genome into a double-stranded DNA, and then it can be integrated into the genome. Now, before I get into the mechanism, which I think is really complicated, you know, when I first started learning about this mechanism, well, you know, I took a virology course in graduate school, and I'm sure I went over it back then, but that was like 15 years ago or 20 years ago, so I don't remember much about that course. But so I've relearned this so I can teach it in molecular biology, and it's rather confusing. It's rather confusing, but I think I figured it out, and I think it's really interesting. Confusing, but interesting. So what I first want to go over is, let's say, what HIV-1 would look like in the genome, in the human genome. HIV-1 is a human virus, and when it infects a human, it integrates into the genome. So let's take a look at the HIV-1 DNA. So what would it look like in the genome? And we can also refer to this as the provirus. So when it's integrated into the genome, it's considered to be in the proviral stage, or it's the provirus. Now, because of the way HIV-1 integrates into the genome, it creates between 4 to 6 base pair target site duplications. So on either side of the provirus, we'll see 4 to 6 base pairs of identical sequence. And this is the target site duplication, and we saw this with the P element also, and insertion elements when we were talking about transposons. Now, on this side of the HIV-1 DNA, we're going to have 3 sequences referred to as U3R and U5. And this is going to be repeated on the other side. So U3, R, and U5. And, you know, perhaps the better way to describe these are the long terminal repeats, LTRs, of HIV-1. So they're identical sequences found on the left and right sides of the HIV-1 DNA provirus. Now, we saw some transposable elements that had terminal inverted repeats on the ends. So these are not inverted, these are direct repeats. So the exact same sequence at the beginning and the end of this molecule. And they are typically referred to as long terminal repeats, LTRs. Now in here, we're going to have a lot of different genes, and I'll go over those in a different video. So in here, meaning between the LTRs. So now the DNA is not incorporated into the virion. What's incorporated is an RNA. So the RNA genome is incorporated into the virion, or the infectious particle. And what you're going to see, what I want to point out, is that the transcriptional start site is right here, say after the R3 and before the R. So I'm going to put this little arrow to say this is where transcription starts. So RNA polymerase in the human cell will initiate transcription from right here, transcriptional start site, going this way. It's going to go all the way down, and the poly A addition site is going to be between this R and this U5 right here. So we'll have a poly A tail, and this is going to be, we'll call this one the HIV-1 genomic RNA. and it's about 10 kb long, and it's single-stranded, and it's probably capped. I don't know for sure. I'm going to get to that later in a different video, but it's probably capped because most RNA molecules, when they're transcribed by RNA polymerase II, they have a 5' cap, which is 7-methylguanosine. So now you might notice, because transcription started here and ended here, the very beginning of the RNA molecule will have the R region and the U5 region, but it doesn't have the U3. So the 3' end of the RNA molecule has a U3 region and the R region, but it doesn't have the U5. Now I want to point out a couple more things. So this side, after the 5' UTR right here, there's also going to be what we call a primer binding site, so PB. And over here on this side is going to be what we're going to call the PPT for polypurine tract. And those are going to be important for the mechanism by which this RNA molecule is turned into a DNA molecule that can be integrated into the genome. So, and I'll put those down here, PB and PPT. Now, okay, so this is an RNA, and actually 2 of these RNA molecules are going to be put into the virion, the infectious particle. That infectious particle can go and infect another cell. And what we're going to see is that one of the important proteins encoded by these, one of the HIV-1 genes is reverse transcriptase. So RT for reverse transcriptase. Now this reverse transcriptase, the HIV-1 reverse transcriptase, has a number of different properties. One is that it has a DNA polymerase property from an RNA template. So it can make DNA from an RNA template. And that's what we've gone over in the past. RNA reverse transcriptases use RNA templates to make DNA. So it also has a DNA polymerase from a DNA template. So it can also use DNA, single-stranded DNA, as a template also. So it can do both. It can use both RNA as a template and DNA as a template. And another activity is it has an RNase H activity. So RNase is an activity that degrades RNA, so it degrades molecules of RNA into individual ribonucleotides. So RNase H is a specific type of RNase activity where it degrades RNA when it is in a hybrid molecule with DNA. So it degrades RNA in an RNA-DNA hybrid. So if this is a double-stranded molecule right here, if one strand is RNA and the other one is DNA, well, our RNase H activities, what they do is they remove the RNA. They just degrade the RNA into individual ribonucleotides, leaving the DNA intact. So all of these properties are going to be used by the reverse transcriptase to turn this RNA molecule into a DNA molecule that can be integrated into the genome. So now how does that work? So this is where it gets a little confusing. Now I have some diagrams made for us already. Now, okay, what am I showing here? I'm showing this RNA molecule right here. So this is just the HIV-1 RNA that's been transcribed, incorporated into the infectious particle. Now, it needs to be turned back into a DNA so it can be inserted into a genome, you know, when that virus infects another cell, when that virion holding this RNA molecule infects another cell. And like I said, the virion has 2 of these. But those are, we don't really need to consider. We only need to focus on one. I left the poly A tail out because it's just confusing. We can ignore that. And I am diagramming just the R. This is the U5 region. I left the 5 out for some reason. The primer binding site, the polypurine tract, I just put PT in here instead, the U3 region, and the R region. Now, the first thing that happens, see this PB? Well, we're going to need a primer in here for reverse transcriptase to come in here and start making DNA. And the primer is really interesting. So it turns out it's a transfer RNA from the host. In the virus itself, the virion may actually have some of the transfer RNA from the host inside the virion. And this transfer RNA, the 3' end of the transfer RNA, binds to the primer binding site. And it is the primer to start this reverse transcription process. And what is the reverse transcriptase that does this? It's the HIV-1 reverse transcriptase. So some of this reverse transcriptase is going to be in the virion with the RNA. So, okay, so how does this work? The primer binds here, and this binding is really inefficient. So it's really inefficient. But once it gets started, I guess the reverse transcription process is efficient. After this tRNA primed reverse transcription takes off, then it's a rather efficient process. But starting the process is thought to be inefficient, which you can probably imagine using a tRNA to initiate the reverse transcription. So the reverse transcriptase activity is going to start here. This is a 3' end, so it's going to go this way, right here, using the RNA, the 5' end of the HIV-1 genomic RNA as the template. So in the rest of these diagrams, I'm going to show the tRNA in black, the RNA nucleotides in red, and the DNA nucleotides in blue. So after synthesis from the tRNA primer, we have this part of the HIV-1 RNA turned into DNA. Now, okay, something interesting happens here. So remember that RNase H activity? So now that can be used to degrade this RNA part right here that is in this RNA-DNA hybrid, right? So we have a DNA here, RNA here. So the RNase H activity of reverse transcriptase can be used to degrade this. So, okay, so we removed the ribonucleotides that were associated with R and U5 in the RNA. And we have the DNA of R and U5 intact down here. Now, another interesting thing happens, and there are a couple of models to explain how this is possible. But for our purposes, we just need to know that this R region here is complementary to this R region right here. So this whole thing right here could be moved down to here. And you know, it may involve some sort of folding, right? This just folds over. You can anneal this R to this R because they're complementary. So in the next diagram, I'm just going to show this moving down to here. So this R right here, because it's complementary to here, I'm going to show this annealed down here. Okay, so now we have this annealed, and this 3' end, this ends right here. So now that we have these R's annealed, this is a 3' nucleotide right here, right? 3' OH group. So the reverse transcriptase can now synthesize in this direction using this RNA molecule up here as the template. Another reverse transcriptase can use this 3' OH right here to synthesize this way. And that's what we have here. So now we have DNA right here. Oh, I didn't show synthesis here yet. I'm going to show that next. So we have DNA synthesized this way, right here. Ooh, I didn't, okay, I'm sorry. I left something out. No. I made all these diagrams, but I didn't practice this yet. So, okay, we're synthesizing this way first. We're not synthesizing this way yet. I'm not sure why we didn't do that. But what I want to point out is, it could be because there's a poly A tail here that we need to get rid of. That could be why we haven't synthesized in this direction yet. But I'm going to, hopefully I didn't confuse you too much. Let's back up a little bit. This R is annealed to that R. We're synthesizing this way, okay, using this RNA molecule as a template. Okay, we have that synthesized. Now, we have an RNA molecule hybridized to a DNA molecule down here. We also have it right here and right here, essentially all along here. So the RNase H activity of reverse transcriptase can now chew all of this away, leaving us with just DNA. But there's something interesting that happens here. So this PPT, polypurine tract, has lots of G's and A's in the RNA strand here. And there's something neat about this sequence that keeps the RNase H activity from degrading just this segment right here. It doesn't degrade this right away. So what happens is it degrades all the other RNA except for this right here. So I'm showing all the ribonucleotides just as, you know, being chewed up by RNase H. So what that leaves us with is something like this. Okay, now the reverse transcriptase can use this 3' OH right here to synthesize in this direction. And okay, keep in mind that even though this is in black, this is a tRNA. So these are RNA nucleotides. The blue is the DNA nucleotides. Okay, so reverse transcriptase is going to use this 3' end to synthesize this way. And what we end up with is something like this. So now we've copied the U3, the R, the U5, and the PB region right here. And you can probably see, if you're paying close attention, that this is tRNA sequence, right? So the PB region were the sequences of the tRNA that were complementary to the PB site of HIV-1. So the RNase H activity of reverse transcriptase can chew this away now and release that tRNA, which is going to be no longer functional, even for a tRNA. So it's going to chew away this part, and it won't be annealed with this PB region anymore. So, okay, so we're left also, what I wanted to point out is that researchers are still kind of trying to figure this out, is why is it after this PPT right here, which was the RNase H activity of reverse transcriptase was initially unable to degrade. After it's used as a primer, it finally becomes susceptible to degradation. And whether or not it's a different RNase that takes it away, or it's that RNase H activity of HIV-1 reverse transcriptase that can finally chew it away once this piece is synthesized, I'm not really sure. But after it serves as a primer, it can be removed by an RNase H activity. So we're left with something that looks like, well, after we remove that PB, that complementary site of the tRNA that's complementary to this PB site right here, and the ribonucleotides of the PPT, once those are removed, we end up with something like this. Now, this piece right here has to be moved down to here. And again, there are some different models that can explain how this is done, but it might be that this just curls around and we get this PB annealed to this PB, but we don't even need to worry exactly about how that happens. what I want you to see is that what I'm going to do is I'm just going to shift this piece right here all the way down to here. So this PB right here, which is complementary to this PB right here, so these are annealed. This PB is annealed to that PB right there. Okay, and I have these annealed. And now you can see we have a 3' end right here and a 3' end right here. So the reverse transcriptase, which has a, remember it has a DNA polymerase using, it can synthesize DNA using DNA as a template. So this 3' end can be extended using this DNA molecule as a template, and this 3' end right here can be extended using this DNA molecule as a template. And if you're paying attention now, what you can see is that, oh, we have the U3 end over here, and now we have U5 over here. So after those ends have been extended, we have that complete HIV-1 sort of genomic DNA sequence, right? That has the complete LTR on the left, going from U3, R, and U5, then followed by PB. Then over here we have the PPT, followed by, again, the LTR. So U3, R, U5. Now this is ready to be integrated into the genome. And what will that do? I'm going to go over the integration process in the next video, but that will lead to target site duplications, and it will give us the LTRs into the genome with the rest of the viral sequences. Now, okay, so again, just to summarize, so when this provirus is transcribed, the RNA does not have the U3 sequence at the 5' end or the U5 sequence at the 3' end, but because of the way this molecule is reverse transcribed before that DNA molecule is integrated into, say, a new host genome, because of that mechanism, the U3 is added back to the 5' end and the U5 is added to the 3' end. Okay, I hope you understood that video or overview of HIV-1 reverse transcription, and we'll see you in the next video when I talk about integration of the reverse transcribed HIV-1 into the genome.